Welcome to Shaping Science, a collection of videos from voices that are shaping science today. I'm Yuni Wang from Drug Discovery News, and today we're joined by Jasmine Trigg from Sartorius. We'll be exploring how live cell imaging is transforming the way we approach neuroscience research. From groundbreaking discoveries to real-world applications, we'll dive into the ideas and innovations that are shaping the future of science. So without any further ado, let's get into the science. Hi, Jasmine. How are you today?
Hi, I'm good, thank you. How are you?
I'm good, thank you. Firstly, when studying neurodegenerative disease like Alzheimer's or Huntington's, why is it so important to monitor neuronal activity over extended periods of time?
So I think the first thing to consider is and kind of really highlight is that in neuroscientific research, it's the kind of one of the main challenges we face is really intrinsic to the nature of the neuronal cells that we're using. So these cells are kind of difficult to obtain, they're costly, and they're quite sensitive to handling as well. And these cells will often involve long or complex protocols, especially if we're looking at mature cell types. So what researchers need to be able to do is maintain the cultures for long periods of time and analyze changes that occur. So traditional techniques used to assess neuronal activity are typically endpoint ones. So these lack temporal data. They also have additional limitations, such as increasing the chance of damaging cells or introducing artifacts. And these measurements are derived from a limited number of cells. So this means they don't kind of mimic the true network activity that we see with neurons or their kind of connectivity between them. So if we look at this in the context of neurodegenerative diseases like Alzheimer's and Huntington's, we're really modelling disorders of slow progression and high plasticity. So it's really critical to monitor the changes that we see over time. Yeah, live cell analysis allows us to get kind of dynamic and cellular interactions and measure the same neuronal population and kind of really get some insights into the subtle changes that occur.
How do you actually capture and monitor spontaneous neuronal activity over days or even weeks?
So we've developed the InKsight live cell analysis system, and this is a imaging and analysis system that resides inside a standard lab incubator, and this allows us to acquire phase and fluorescence images of live cells in a non-invasive manner. Alongside this, we established the InKsight neuronal activity assay, and this is a end-to-end solution that allows for direct long-term measurements of spontaneous neuronal activity. And what this allows scientists to do is really look at get a functional insight into neuronal cell models. So we have validated protocols for this assay, and we use a reagent as part of it, which is the InKsight Neuroburst Orange lentivirus. And this is a genetically encoded fluorescence calcium sensor. It's non-perturbing to the cells, and it's also neuron specific. So it means we can look at neuronal activity in co-cultures as well. As part of this, we have specific acquisition mode and integrated analysis, and this allows us to look at longitudinal changes for every active object within the microtiter plate. And this means we can analyze thousands of cells in real time from days or kind of weeks and months as well. This kind of overcomes the limitations that we see with more conventional approaches. We've got this kind of temporal nature, it's miniaturized and non-perturbing, but it is also non-exhaustive, so it is still complementary to downstream techniques such as high-con imaging or flow, and it can really kind of aid in assessing the kind of best time points to perform these assays as well.
What new insights into neurodegenerative disease mechanisms have you or others in the field uncovered through this long-term live cell observation?
So a couple of years ago, myself and a former colleague, Susanna, we used live cell analysis to investigate neuronal activity in Alzheimer's disease models. And here we were particularly focusing on a protein called tau. So Alzheimer's is characterized by the abnormal formation and aggregation of proteins in the brain, and this includes amyloid beta and tau. researchers kind of a number of hypotheses underlying the disease progression, and one suggests that in Alzheimer's, tau becomes kind of hypophosphorylated and it loses its normal ability to bind to the microtubules and stabilize them. And instead, we see that this protein can accumulate, and this kind of in turn can lead to some of the kind of pathological hallmarks of the disease. So to investigate this, we co-cultured rat cortical neurons and astrocytes, and the neurons were expressed in the neuroburst reagent, and then we imaged them on the InKsight every 24 hours. So what we could do is, once we'd seen in the assay that mature networks had formed, we then treated them with the aggregated tau. And after analysing the calcium fluctuations, we found that if we compared these to vehicle, tau treatment decreased the number of active objects and their mean intensity as well. And they didn't affect the correlation. So within the assay, correlation's a measure of network connectivity, so kind of how well the neurons are all kind of bursting together at the same time. The preliminary data suggested that the neuronal activity was perturbed in the presence of tau, and really highlights how we can use the assay to look at the effects of disease-related proteins on neuronal activity. More recently, we used live-cell analysis to monitor neuronal activity in iPSC-derived models, and these models provide a kind of a more translational system for investigating both development and disease. So in this example, we used healthy and Huntington's disease human iPSC-derived neurons, and these were expressed in neuroburst. And we looked at these either in monoculture or in co-culture with iPSC-derived microglia. These were then monitored in the InKsight for up to 45 days. So when we look at the results, these indicated that for both healthy and diseased neurons, the functional profiles altered between mono and co-cultures. So we saw that in the co-cultures, there was an increased number of active objects, and we saw kind of greater correlated activity between the neurons firing as well. And this really indicates a more mature and stable network in the presence of microglia. Interestingly, when we then compared the activity of the healthy and the diseased co-cultures, we observed similar levels of correlation, but we saw that for the diseased co-cultures, there was an increased duration of calcium events. So essentially, the neurons were kind of firing, but the burst was a lot more prolonged to what we saw for the healthy neurons. And this indicates a disruption in the neuronal activity in this disease model.
What excites you the most about the future of neuroscience research using live cell imaging tools?
So neuroscience, I think, is quite a dynamic field. There's a lot of different exciting things about it. I think for me, one thing I find really exciting is just the continuing advances in iPSC development. So as I mentioned, we kind of have worked a lot of these recently, and these iPSC-derived models really offer the potential for kind of easy access to healthy human or patient or disease-specific cells. And this gives us more representative models through which we can look at neurodegenerative pathways and also test out therapeutics. I think the continued development of these will limit our reliance on kind of more subpar animal models, and hopefully reduce the number of drugs that we see that kind of fail when they get to human clinical trials. Kind of related to this, I think another really interesting area within stem cell research is cell-based therapies. So this is kind of very much still in its infancy, but I think it's kind of been acknowledged. It has potential to be a really powerful and innovative strategy for treating neurodegenerative diseases. So in theory, we can differentiate stem cells to desired cell type, like a specific neuronal cell. We could then transplant them into the brain and then replace any lost or damaged cells, ultimately kind of trying to restore any defects that were there. However, I think for clinical trials and a path to commercialization, we really need a detailed understanding of the cells and their critical quality attributes. Probably a more short-term and attainable goal is maybe looking at how stem cells could deliver therapeutic factors and look at kind of preventing the disease progression.
What's the most surprising thing you have observed when monitoring neuronal activity?
Yeah, I think I'm not sure about surprising. I think it's kind of an expected part of the assay. It's kind of what it's meant to do, but I still do get kind of excited whenever I see neurons start to fire as part of when I'm setting up an assay, even now after doing it kind of multiple times. So it's really nice. You kind of see the neurons when they kind of start to express the lentivirus, they're kind of like subtle stars, and you can see them kind of gently firing. And then as they kind of mature and connect, the objects get a lot brighter, and we see these big kind of bursts of correlated activity. They're kind of like fireworks when you see them all going off kind of at the same time. And it's really kind of nice and exciting to be able to visualize this as a researcher and just see how your kind of model's progressing and any changes throughout that development as well.
Thank you so much, Jasmine, for joining us today. To learn more about live cell imaging and analysis, please click the link in the description. Thank you for watching. Together, we are shaping science.